Hello everyone and welcome to another recorded lecture of BIO1 online. Today we'll discuss chapter 13, which is on the evidence of evolution. Before discussing chapter 13, I'd like to just assign a brief reading assignment for the next chapter, on chapter 15. I'd like you to read the first chapter and introduction to the book Sapiens by Yuval Noah Harari. This book was released a few years ago, but still remains a very popular bestseller. It's a fantastic book that gives us a brief history of humankind and tells the story of what makes humans special, or are we really that special after all? So that's for next chapter. So now we can start talking about the evidence of evolution. In Darwin's time, some scientists suspected that Earth was hundreds of millions of years old, but no one could have known that Earth was formed 4.6 billion years ago. And scientists use what's called the geologic timescale to divide the history of Earth into eons. We have several eons. We have the Hadean eon, the Archean eon, the Proterozoic eon, and then we have eras. So eons are divided up into eras. And these eras are divided based on evidence of biological events, such as mass extinctions, or geographical events, right, like an ice age or some kind of volcanic eruption. And the earliest evidence of life is about 3.8 billion years old. So we could say that life on Earth, and these again are the first cells, arose on Earth 3.8 billion years ago, and life has been diversifying ever since. And the millions of species that are alive today did not just pop into existence all at once. They're the result of continuing evolutionary change that started with the primitive organisms that lived billions of years ago. Right, so starting at around 3.8 billion years ago, that's when life first starts. And ever since, we see this huge diversification of life. But everything shares a common ancestor. And now we have a framework to explain this diversification. We know about natural selection. Right, changes in the environments on Earth selected for changes in body structures and molecules that provided an advantage for survival and reproduction. So basically, Earth continually changed, the environment keeps on changing, and those changes accumulate, they put pressure on organisms, right? To basically, if they have what it takes to survive, then they can pass on those genes to the subsequent generations. And those changes accumulate over time, producing the variety of organisms we see today. So it's very hard for the human brain to conceive how a whale and a palm tree can have a common ancestor. But we have to remember this happened over billions of years, and the human brain is very limited in its conception to begin with. So we have to just get over the fact that we might not be able to really conceptualize some of these amazing biological concepts. Right? But we now have a framework that makes a lot more sense than saying, oh, they just were created on a certain day and they just came to exist and they never changed. Right? We know from evidence that organisms always change, and the environment is what specifies those changes. So we'll discuss today the evidence for evolution. Right? I don't want you to just accept that it's true. I want you to use your reasoning and believe it if there's enough evidence. So there's abundance of evidence that supports this theory. Mostly we'll discuss how fossils and paleontology tells us a very clear story of our evolutionary past. We'll also discuss comparative anatomy between organisms, and we'll analyze molecular sequences. And between fossils, comparative anatomy, and molecular sequences, we can paint a picture of how species are related to one another. And it's important to know that there's no way to get an 100% clear picture of what took place on Earth over the past billions of years, right? It's almost like testing evolutionary theory and evaluating common descent is kind of like solving a crime, right? So we have to piece together the evidence that we do have and try to make a story out of it. And if any piece of evidence contradicts the story that we have to basically throw the story away and start over. Right? If you convict a certain suspect of something, and then there's a piece of evidence that exonerates that suspect, you can't still convict the suspect. Right? So in that way, we can use our evidence to the best of ability to come up with a perpetrator. But there's no way to see 100%. So, for example, Darwin looked at evidence, not at a crime scene, but on the Galapagos Islands, and he saw finches and how they had different beaks that were suited to their natural environments. So that helped pave the way for natural selection. Right? And that could help explain how we come from a common ancestor. Right? Similarly, fossils are literally like crime scene evidence. They leave behind like snapshots of clues to past life. And that really is what allowed Darwin to make some of his breakthroughs, was the fossil record. And the best explanation is one that is consistent with all the available evidence, right? So the theory has to be consistent with the fossil record, but also with DNA sequences and with comparative anatomy. So we'll start by talking about fossils first. So paleontology is the study of fossil remains. And by definition, a fossil is evidence of an organism that exists for more than 10,000 years. So all fossils are at least 10,000 years old. And like I mentioned, fossils kind of provided the original evidence for evolution and allowed Darwin to make some of his hypotheses. The oldest fossils are over 3 billion years old. Right? And again, when we think of fossils, sometimes we just think about, like this Triceratops over here. But we have fossils from plants, like this leaf over here. This is actually a flowering plant that lived in China more than 100 million years ago. This is petrified wood. This is a dinosaur egg that's been preserved for over 190 million years. Even turtle feces could be fossilized. Here you see a fish that's been very well preserved, or trilobites, which were arthropods that became extinct 250 million years ago. So this is, again, you can't really argue against these kinds of fossil records. And we'll talk about how we come up with the dates for these fossils. So we could actually figure out how old the fossils are and conclude when certain evolutionary events took place. So this way we can document the history of all life on Earth. So a fossil often forms when an organism dies, becomes buried in sediment, and then becomes chemically altered. And this is the case for the coal and the natural gas that we use for energy today. So these were actually once living organisms that are fossilized. That's why we call them fossil fuels. So really short, hundreds of millions of years ago, this is before the dinosaurs, there were many giant plants that died in swamps. And again, these plants are made up of cells, which are made up of biomolecules. A lot of them contain energy in them. Right? Remember that all these structures have energy. But these were, over millions of years, they were buried under a lot of water and dirt. And a lot of that heat and pressure turned all of the dead plants into coal. So all of the energy that these plants once obtained from the sun through photosynthesis basically got trapped and reconverted into a different material we call coal. But there's still energy to be used. We can still break the bonds in coal and burn it to produce heat. So again, this is kind of talking about how in thermodynamics we can convert one form of energy into another. Same idea for natural gas formation. So beneath the sea floor, we now have the decomposed remains of a lot of these sea plants and animals that were converted by a lot of the heat and pressure into oil and gas. So we can drill down through the layers of rock to reach all of the oil and gas deposits that are, again, hundreds of millions of years old. So those are fossil fuels. So now I want to discuss a few ways in which fossils may form. One way, like we've just seen in the case of fossil fuels, is compression, where some kind of organic matter like a leaf sinks and sediment covers the leaf. And once that sediment compresses, it forms into rock. So you're compressing this leaf into rock, basically. So you're compressing it. That's how natural gas and coal was formed. Another way to preserve a fossil is with petrification. And in petrification, minerals replace those that were found in organic matter left in the decaying organism. and that literally turns into stone. So, for example, this skull, this bone was buried underneath dirt, and water containing minerals was able to seep through into the bone. And those minerals replaced minerals in the bone and turns it into stone. So it literally gets mineralized. And that's how petrified wood forms. So this is a petrified skull. So that's petrification. Another way to do it is with impression. An impression forms when an organism presses against soft sediment, making an impression in like mud. And then as the animal's body decays, it still leaves an impression. Right? So you can actually see the scales of this dinosaur. Another way we can have a fossil be formed is with a cast. And a cast is kind of just like what it sounds. What happens first is that an animal will die, and then it will form, just like over here, it will form an impression. But then that impression gets filled with mud. And once that mud hardens into rock, you have an actual cast of what that organism originally looked like. So it's like a 3D protrusion that tells you what the shape and structure of that organism was. Finally, and the most rare way to fossilize an organism is with intact preservation. So sometimes tree resin is very sticky and it can trap some plants or animals, like this insect over here. And then it can become basically preserved for millions of years. So this was kind of what Jurassic Park was based on. They said that it's not really true, but they were able to extract DNA that was found in a mosquito from dinosaur. That mosquitoes once bit from dinosaurs, and the mosquito got trapped in amber resin. I think that's the story anyway, so don't hold me to that. But I'm pretty sure that's the premise of Jurassic Park, is this fossilized mosquito was preserved intact, and it had some bit of dinosaur DNA that was able to then be used to resurrect the dinosaurs. So those are ways to make fossils. So as informative as fossil history is, it's still not a complete picture. The truth is, most organisms fail to fossilize. It's a rare occurrence for fossilization to occur. So we're missing out on most of what happened on Earth. More so, erosion and movement of the Earth's plates probably destroyed most of the fossils we see. All right, so those that did fossilize were probably destroyed, or more likely are inaccessible to us humans, or we haven't found them yet. So even though fossil evidence is incomplete, we can still get lots of invaluable information from the fossil record. For example, these marine fossils were found in Oklahoma. And again, the only way that this could have been the case is that this area of Oklahoma used to be under an ocean. And we now know that that's true. So Oklahoma used to be a shallow ocean. And we could basically infer different ways that Earth's plates might have moved over time. So we can determine the age of a fossil in a couple of different ways. And dating fossils will give us clues about the timeline of life's history. So one way we can determine the age of a fossil is using relative dating. And relative dating places a fossil into a sequence of events without assigning it a specific age. So it's relative. And relative dating uses the principle of superposition, which assumes that the lower levels of rock, or the lower layers of rock, have older fossils than the newer layers. So theoretically speaking, I would be seeing something down here that is a lot more ancient than something up here. So just by knowing that, if I see, let's say, some organism up here and it has different features than an organism down here, I would assume that the newer features were more recently acquired, right? Not the other way around. If I saw something down here with different features, I would assume that, oh, it was originally like this organism up here, but then it lost those features. I hope that makes sense. So just by looking at different layers of rock, we can basically get a timeline of what happened first. And we can basically piece these pieces of evidence together to stitch together a story. But that's, again, not as direct as we want. So relative dating just places fossils in order from like oldest to most recent. However, absolute dating is a lot more specific. So researchers could use absolute dating, or also called radiometric dating. And this measures the amount of an isotope in a fossil to calculate how long ago the organism died. So whereas relative dating basically tells you this rock is older than this one, absolute dating can tell you how many millions of years old each rock is by measuring the amount of an isotope in a fossil. And if you remember, an isotope is an alternate form of an element with a different number of neutrons. So I'm going to try to explain radiometric dating very simply at first. So throughout life, organisms accumulate two forms of carbon from the environment. Most of the carbon we use is the stable carbon-12. But there's also a version of carbon called C14, which is a radioactive isotope. A radioactive isotope is one that decays at a very predictable rate. So these radioisotopes are unstable. So they basically decay at a very steady rate. So it's important to know that while an organism is alive, there's a constant rate of C14. So organisms will accumulate both versions of carbon, carbon-12 and C14, but there's a constant rate of C14 in their tissues while they're alive. But after the organism dies, no more carbon is added, and C14 will constantly decrease. Right? As there's no more incorporation of new carbon, we know that the carbon-14, since it's a radioisotope and is not very stable, it will start to decay. And we happen to know that C14 decays at a very steady rate, and we know that in every organism, half of the C14 is lost every 5,730 years. So we know that after the death of an organism, half of the C14 is lost every 5,730 years. We can therefore say that the half-life of C14 is 5,730 years old. That's a very important term, the half-life. And every radioactive isotope has its own half-life, right? That's the rate at which it decays, and it's unchanging. So the half-life is the time it takes for half of the atoms in a sample to decay. So we're looking at C14. So the half-life is the time it takes for half of the C14 to decay. So after one half-life, after death, after 5,730 years, if there was 100% originally at the time of death, then after 5,730 years, there's going to be 50% of that original amount of C14. But now this is a little tricky. We keep on halving it again. So after another 5,730 years, we have this amount. So it's going to get down to 25%. After 3 half-lives, right? So that's over, 16,000 years we're talking about now. Then we're going to get 12.5% of the original C14. So I want you to watch a video to help explain it, and then we're going to go into more details. So here's a brief video contained in the ebook.
After an organism dies, it might become a fossil. Scientists often use fossils to learn how species are related to one another and how long ago they lived. Dating fossils yields clues about the timeline of life's history. Scientists know that fossils buried in older rock layers are from ancient organisms, and that newer rock layers contain more recent fossils. But what tools do scientists use to determine a fossil's age more precisely than that? Radiometric dating assigns an approximate age to rocks and fossils. One well-known type of radiometric dating is carbon dating. In this tutorial, we'll take a look at how this process works. Let's start by looking at a bone in a living animal. The bone contains organic molecules which are composed partly of carbon atoms. Carbon has multiple isotopes. Most carbon atoms are carbon-12, which has 6 protons and 6 neutrons. But trace amounts of radioactive carbon-14 are also present. This is a hypothetical sample of 18 carbon atoms from the living bone. 3 carbon-14 atoms are depicted in this sample, but in reality, the concentration of carbon-14 would be much lower. Carbon-14 decays to nitrogen-14 at a known rate. While the animal is alive, its ratio of carbon-14 to carbon-12 stays constant, as it continues to obtain both carbon isotopes from its food. However, a dead organism adds no new carbon. Yet carbon-14 continues to decay even after death. The proportion of carbon-14 therefore declines after an organism dies. This fossilized bone has a lower ratio of carbon-14 to carbon-12 than a bone in a living animal. Precisely how much lower depends on how much time has passed since the animal died. Every radioactive isotope decays at a constant rate, which is called the isotope's half-life. The half-life is the amount of time it takes for half of the radioactive isotopes in a sample to decay. Carbon-14, for example, has a half-life of 5,730 years. Since this half-life never changes, we can use the amount of carbon-14 in the bone as a sort of clock to determine how long ago the animal died. In this graph, the y-axis shows the percent of the original amount of carbon-14 remaining in an organism. The x-axis shows time measured in half-lives. Since we know carbon-14's half-life, we know that each segment on this x-axis represents 5,730 years. As we've already seen, the percent of carbon-14 does not change while the organism is alive. The dotted line marks the death of the organism. After death, carbon-14 decays and it's not replaced. After one half-life, only 50% of the carbon-14 remains. After 2 half-lives, 25% remains. After 3 half-lives, 12.5% remains, and so on. We can use a graph like this to estimate any fossil's age. For example, if scientists find a fossil with 25% the amount of carbon-14 as occurs in a living organism, they can conclude that the organism died about 2 half-lives ago, which is equivalent to 11,460 years. Carbon-14 is good for dating fossils up to 40,000 years old, but many fossils are much older. Luckily, carbon-14 is not the only isotope used in radiometric dating. Potassium-40, for example, decays to argon-40 with a half-life of 1.3 billion years, so it's useful for dating much older fossils and rocks.
Great. So hopefully that clarified a little bit, but let's go into a little more detail and reiterate some of those concepts. Right, we said that we can date a fossil with absolute dating by measuring a radioactive isotope. And while alive, there's a steady amount of incorporation of the radioactive isotope as it decays. But after an organism dies, the amount of the radioisotope decays at a very steady rate. So if we found a fossilized woolly mammoth, for example, we could measure the amount of C14. And we can infer, we could look at a living organism, and we know the exact proportion of C12 to C14 that should be in a living bone. And we can measure the amount of C14 that's in the dead bone and infer how long ago this must have been alive. So there's a simple equation that we use to figure out how long the fossil is. The number of half-lives elapsed is n, where the fraction of isotope remaining is 1/2 to the n. Let me explain what this means a little bit more. After 0 half-lives, right? We can say that this is, well, let's use 1. After 1 half-life, let's plug in 1 for n. After 1 half-life, there is 1/2 of the isotope remaining, right? We could just plug in number of half-lives elapsed. So after 1 half-life, 5,730 years in the case of C14, there should be 50% or 1/2 of the isotope remaining. After 2 half-lives, let's plug in this 2 for 2, it's 1/2 squared. So after 2 half-lives, there's 1/4 or 25% of the isotope remaining. After 3 half-lives, there's 1/8 or 1/8 of the remaining isotope. So using this equation, you can solve 2 kinds of problems. You can be told an organism or a fossil is this many years old, how much C14 is remaining in it, given a half-life of 5,730 years, or it could be the opposite way. You could be told there is this amount of isotope remaining, how old is the fossil? So let's do a sample problem together. So researchers find a rock containing fossils of ancient microbes. The rock contains 12.5% as much of the radioactive isotope as it did when the rock was formed. If the isotope's half-life is 1.3 billion years, how long ago did the fossil form? So pause here, don't look at the answer, and try to use that equation that we just figured out to figure out how long ago the fossil formed. So let's write down our equation. We know that 1/2 to the n, where n is the number of half-lives, is equal to the percentage of isotope remaining. I'm just writing isotope. So we're given 12.5%. The rock that we found that's fossilized has 12.5%. I hope 12.5% you can easily, in your head, recognize that that's 1/8. 12.5% is 1/8. It's the same thing. And then, so our equation becomes 1/2n equals 12.5% or 1/8. I'm putting it in fractions because it's easier. Now, it tells you, so we can figure out, we can solve for n, and that tells us the number of half-lives. Right? And n must be 3 to solve this, right? 2 cubed, 2 times 2 times 2 equals 8. So n equals 3. And that is basically our answer. It's 3 half-lives have passed since this fossil was alive. And you're told the half-life is 1.3 billion years. So you have to multiply 1.3 billion years times 3, because it's 3 half-lives. Right? So, and again, we could look at this graph. If there's 12.5% remaining of the isotope, that means there's 3 half-lives passed. And in the case of this radioactive isotope, if it had a half-life of 1.3 billion years, that means that the fossil formed 3.9 billion years ago. And that stands for billion years ago, if I didn't say that already. Right, so we multiply 3 half-lives times 1.3. So pause here and try to answer this question. Which rock layer should have fossils with the most carbon-14? And the answer is A. Right, because fossils up here died more recently, and those would have the most carbon-14. These would have the least, because those died longest ago. If the half-life of a radioactive isotope is 5,000 years, how much of the isotope would still be there after 15,000 years? So pause here and try to figure that out. All right, so again, we could use our equation. 1/2n gives us the percentage left. In this case, we're trying to figure out the percentage. And we're given n, because we can realize that if 5,000 years is 1 half-life, 15,000 years is 3 half-lives. Right, so that n would be 3. And again, here we have right? We plug in 3 for n, we're going to get 1/8, or 12.5%. So it's just that simple equation you have to remember. Let's do one more. Researchers used a radioactive isotope with a 25,000-year half-life to date a fossil to 100,000 years ago. How much of the fossil's isotope is remaining? So again, 25,000-year-old half-life, 100,000-year-old fossil. That means 4 half-lives have passed. So with 4 half-lives, right, 1 over 2 to the 4, we know that it's going to have d 1/16 of the amount of the isotope. And again, you should be able to do this question the other way. If I said, researchers used a radioactive isotope with a 25,000-year half-life, they found that the fossil contained 1/16 as much of the isotope as does a living organism. How old is the fossil? So you have to be able to realize, right, figure this out to get to 100,000 years old. And I recommend looking at the back of the book, the back of the chapter, for some more sample problems. So now let's move on to talking about biogeography. And biogeography combines the studies of geography and biology to study the distribution of species across the planet. And Earth's geography has always been changing. It's not always the nice 7 continents that we know today. So over the past 200 million years, the continents have been shifting, and that affects where organisms can live and where they can migrate. And plate tectonics explain why continents drift. This is called continental drift. Continents literally drifted away from each other over the past 200 million years. At one point, all the continents were joined into one supercontinent, Pangaea. And because of plate tectonics, which explains how different tectonic plates beneath Earth's surfaces move and create drastic shifts, they can create mountains or deep sea trenches. Right? So because of the shifting of plate tectonics, the Earth's landmass can actually move. And Earth's landmasses are actually moving as we speak. And we can actually measure the rate of continental drift. Right? So earthquakes and volcanoes are evidence that Earth's plates continue to move today. So now we know that continents drift about 1 inch per year. And this could help explain some very unusual things. So we found some marine fossils in the Himalayas, in the Himalayan mountains. So how could that have ever been, right? How could the Himalayas have marine fossils, right? How could they have made it 3,600 meters above sea level? Well, because of plate tectonics, we know that the Himalayas were actually once an ancient seafloor. And that was able to rise due to the shifting plates, and that lifted all those marine fossils toward the sky. So again, biogeography and plate tectonics talks about how land masses of Earth constantly move, and that helps explain a lot of the fossils that we find. So let's watch a quick animation. So this shows the drift over time from Pangaea, which was one big supercontinent, into today, how we have the 7 continents. Again, let's just go through that slowly. Right. So that is continental drift. And according to the theory of plate tectonics, it is the forces deep beneath Earth's surfaces that contribute to the movement of these continents. And this dramatically affects life's history, right? As oceans shift and mountains appear and disappear, and different connections between continents merge and vanish beneath the seafloor. So like I mentioned, biogeography is the study of species distribution across the planet. And the locations of different fossils helped explain how Earth's continents were once one big landmass called Pangaea. So because fossils of this Triassic land reptile is only found in parts of South America and Western Africa, we could put together pieces of the puzzle to know that these two land masses were connected. And of course, that's not evidence enough to know. It's all these pieces taken together shed light that there were all these different evolutionary events that were marked by changes in the continents. Right. So again, this other freshwater reptile is only found in parts of southern Africa and southern South America. So these must have been connected, close enough for a freshwater reptile to go from one landmass to another. So Alfred Russel Wallace, who independently discovered natural selection alongside Darwin, was investigating something very unusual in the Malay archipelago. So he noticed that there was this imaginary line, now we call it Wallace's line, where on one side of the line, west of the line, all of the animals were very much like Asian, those that were found on the Asian continent. So there were a lot of tigers and elephants, those kinds of creatures. But on the other side of the line, there were more like Australian creatures. So he wondered, how can there be, what was preventing these, or why was there such a stark difference between one side of the line and the other side of the line? And it turns out that there's a deep sea trench in between these two, in between this line. And that prevented any of the animals from this side from migrating over here and vice versa. So because of this deep water trench that separated the islands, there was no evolution or no migration that can happen. So that basically allowed evolution to produce a unique variety of organisms on each side of Wallace's line. And again, because of this observation that there was such a difference in the creatures on one side of the line than the other, that informed some information about geography, right? Because of the biology difference on both sides of the line, that told Wallace, maybe there's something about the geography of this line. And sure enough, we know now that it's a deep water trench that separates islands. So over millions of years, these organisms on either side of Wallace's line have been evolving separately. So way back in the beginning, we said that we could infer that all organisms share a common ancestor because we're all made of cells, built of the same biomolecules, we use the same genetic code, build proteins of the same amino acids, and so forth, right? So that infers, that gives us enough evidence that we all share common ancestry. We could also look at a larger scale commonality, like anatomy. So birds, reptiles, amphibians, mammals, all have similar bones in their forelimbs. So again, if all these different types of creatures, all these different kinds of animals have a very similar structure of bones in their forelimbs, the simplest explanation, at least, would be that all of these modern vertebrates descended from a common ancestor that had a similar skeletal organization. And that over millions of years, each group gradually modified that skeleton as the species adapted to their environmental conditions. So again, you can say that, again, one argument would be, okay, all of these creatures just independently happen to come up with this arrangement. That's possible, right? But I think it's more likely, and right, most people would agree, it's more likely that all of these organisms share a common ancestor that had some kind of arrangement of bones, and that arrangement of bones got modified, right? That's descent with modification. We all descended from a common ancestor, but along the ride, there were a lot of modifications. So homologous structures are body parts which are similar in different species. Okay, so two structures are homologous if the similarities between them reflect common ancestry. So homologous structures develop differently over time. So what happens is, well, there was once a common ancestor or a parent species that had one type of structure, but then something over time caused the species to diverge. So divergent evolution refers to how a species goes under a change. There's a change where there's a divergence. And on one side, maybe these species modified a structure in one way, and the other half of the species modified their structures in a different way. But they still came from the same parent. So we call those homologous structures. And again, like I mentioned, bones of the forelimb are conserved between mammals. We have the same humerus radius ulna arrangement as a cat or a bat or a whale. Right? So whether you're flying or you're using it to swim, we still have the same arrangement of bones. This indicates that all mammals share a common ancestor, but then depending on our environments, those put pressure on those structures. So a whale would favor a different arrangement of those bones, but yet we still have the homologous bones, I should say. So again, this explains descent with modification. We had homologous structures, right, shared a common ancestor, and then underwent divergent evolution. We call it divergent evolution. They separated over time differently. That's due to something in the environment that favored this divergence. So as environments change, some select So in natural selection, we discussed how as environments change, certain structures of organisms are favored for reproductive success, and thus will hang around from generation to generation. But sometimes, even as environmental conditions change, some structures still persist even if they have no function, even if they're not used at all. So a vestigial structure has no function, but it is homologous to a functional structure in another species. So that's important. So vestigial structures provide information about the origin of species, but they don't really do much for the species themselves. You could think about the word flavor compared in like British or American, right? The British use a U. So if you see the word flavor with a U, okay, it means the same word, but it gives you information about where that word came from, right? It's like, oh, okay, this must have been from somebody who's British, if you see it spelled with a U. I hope that makes sense. But some examples of vestigial structures would be vestigial eyes in this blind mole that burrows. So this mole spends most of its time underground and has no need for an eye, and in fact, it relies on other senses to navigate. But beneath this fur, there still is an eye structure. And that means that this must have diverged, right? There must have been a common ancestor from other mammals that used eyes. But this species just didn't have the use for eyes anymore. Maybe it used more energy than it was worth. But they didn't just disappear from these moles altogether. Another good example is hind limbs in some snake species, like boa constrictors and pythons. So they don't have any limbs, but they do have evidence of little tiny hind limbs. So that might suggest that snakes evolved from reptiles that were once walking on limbs. Another example is pelvises in whales. So ancient whale ancestors from fossils we see have hind limbs and a pelvis, actually, which indicates that whale ancestors, or the common ancestors of whales, once relied on little limbs to walk on land. And even modern whales still have vestigial hind limbs. So if you were to look at a modern whale's skeleton, you would see that there's still remnants of these hind limbs. So apparently, right, their aquatic habitat selected for the forelimbs that were more modified into flippers than limbs. Right, so these are limbs of the ancestor and a pelvis, whereas modern whales have flippers, but there's still you could still look and see evidence of hind limbs, and that gives us again a bigger picture, right? Tells us a story about where this whale might have come from. In humans, we used to think that our appendix is vestigial because you could remove an appendix with little harm. Now that's questionable, right? There might be more functions in the appendix because it might store some healthy bacteria for us that help us in digestion and other aspects of health. But human embryos still have tails. We have a tail as an embryo. We have gills, right? Pharyngeal slits that kind of are only found in certain points of development. But again, that indicates that we must share a common ancestor, right, with other organisms. But we didn't need gills, and we don't need, for example, goosebumps, where other mammals that are hairy rely on goosebumps for warmth and to also appear larger to threaten other species. We still get goosebumps, but it doesn't keep us warmer, and it doesn't keep anybody from attacking us, right? It's just a leftover. So that's what vestigial organs are. They're leftovers. So just because structures look alike or share a common function, it does not make them homologous structures. So anatomical structures are considered analogous, not homologous, but analogous if they appear similar, but do not have a common developmental pathway. So for example, the wings of a bird and the wings of an insect are both used for flight, and they kind of look similar. However, the way that wings develop in birds is completely different from the way that they develop in insects. They're made of different things. They don't have a common developmental pathway. So therefore, we can say that these are analogous and not homologous. So analogous structures result from convergent evolution. So analogous structures are often the product of convergent evolution, which is the independent evolution of similar features in species of different lineages. So this is saying how two unrelated parent species kind of converged and share similar adaptations. Right, that's different from divergent evolution, right? So in convergent evolution, we get similar adaptations in organisms that do not share the same evolutionary lineage. So that's the example, right? So for birds and butterflies, they both needed to fly, but they went about it in very different ways. And then they end up looking more like each other over many, many, many millions of years. But they didn't start off that way. And this is very interesting. There's many good examples of convergent evolution. And I like the example of this body shape. You see a very similar body shape in reptiles, in this extinct reptile, and birds like penguins, and a mammal like a dolphin, or a fish like a shark. We have this streamlined body shape with these appendages that can be used to either swim or, right, penguins can't swim very well anymore, but this has served all these different organisms very well, this body plan. But they don't have any common they don't develop similarly, right? Reptiles and birds and mammals and fish are all very different. But yet, they all illustrate that there was a selective force that favored the development of this body shape, right? And so much so that dolphins and sharks, right, kind of look related, but they're not, right? It just so happened that the marine environment favored the development of structures in fish and in mammals separately, and that just made these two kind of converge and look more similar. An example of convergent evolution is also in these two organisms. This is a salamander, and this is a crayfish. They both are not related, but both separately, kind of not decided, but both environments favored those that lacked pigmentation and lacked eyes. These are not related, but the environmental conditions pressured both of these separate organisms to converge similar adaptations. And this is a really interesting example. So there are two kinds of mammals. We have marsupials, which were once found all over North America, and there are placental mammals. And marsupials were able to prevail in Australia because it was a separate island mass. It's on its own, and there weren't a lot of mammals to compete with those marsupials. Now, this is really fascinating. For several marsupials that evolved in Australia, there is an analogous placental mammal that looks very similar. So as far as burrowers go, a mole that develops with a placenta looks just like a marsupial mole, but they're not related at all. It just so happened that similar environmental conditions pressured these two unrelated individuals to have similar adaptations. Right? So a marsupial mouse and a mouse are not related. But yet both of them ended up looking like this because clearly the environmental conditions in both, let's say, North America and Australia favored, let's say, a long tail and big ears and short fur and so on and so forth. So I hope those examples make sense, right? Convergent evolution is independent evolution of similar features in species of different lineages. So all of these features look similar, but these come from different lineages. They both converged to look more similarly. So to summarize, homologous structures shared a common ancestor and a common developmental pathway, but then underwent divergent evolution. So we said, for example, whales shared a common ancestor with humans. We're both mammals. But because of the marine environment of a whale, they underwent different skeletal adaptations. Their skeleton got modified in a different way than the human skeleton got modified. But if we were to look at the anatomy of a whale and the anatomy of a human, we'd see a lot of similarities because they have a common developmental pathway. But they just diverged over time. So homologous structures share a common ancestor and then underwent divergent evolution. Analogous structures result from convergent evolution of unrelated species. So that's like a bird's wing and an insect's wing are not related. They do not develop the same way, but they both independently developed in both separate species because they were favored by the environment. So in this table, you can see that in mammals, a cat's leg is homologous to a whale's flipper. Right? In mammals, these would be homologous. Just like a praying mantis's leg would be homologous to a water boatman's flipper leg, which is another kind of insect. But a cat's leg is not homologous to a praying mantis's leg. They're analogous. A cat's leg is analogous to a praying mantis's leg. Right? They both are functioning in movement, but a praying mantis and a cat do not develop their legs similarly. These underwent, we could say this is a product of convergent evolution. So the variety of beaks on Darwin's finch is an example of what? So the answer is C, divergent evolution. All of those finches shared a common ancestor. But over time, depending on the environmental conditions on each island, different types of beaks were favored. Right? So they all have a common ancestor, but they diverged differently based on different environmental conditions. But they all share a common ancestor. So the beaks would be considered homologous structures that resulted from divergent evolution. The streamlined shapes of dolphins and sharks evolved independently. The body plan of these two animals are what? So read all the answers closely. And the answer is D, right? They are analogous and they are a product of convergent evolution. So to evaluate evolutionary relationships, we can use what's called a cladogram. And a cladogram is a branch diagram that shows different points in which divergence occurred. So let's say that at this point, these organisms developed wings. And then from that point, they underwent speciation, meaning they developed different species, but they have a common developmental pathway to make wings. We would say that species 1 and species 2 have homologous wings, right? Because they came from the same developmental pathway. Suppose that these organisms diverge without wings in one way, but at a different point in development, wings happen to arise. Well, we would say that species 6 has wings, and those wings are analogous to the species 2 wings because they have the same function, but they do not have a common origin. They have an independent origin. So we could say that species 2's wings is analogous to species 6 because they evolved independently. Species 7 also has wings, but they no longer function. right? The wings are no longer functional at a certain point. So this tells us that the wings are vestigial. And again, if I just looked at species 7 and I see that it has certain wings that look like species 6, but they're vestigial, that tells me that species 7 and species 6 shared a common ancestor. So by looking at anatomy, right, this gives us clues about at which point these different organisms diverged. So I want you to pause here and look at this cladogram very closely. Which two species are most distantly related? So all of these, from reptiles, mammals, amphibians, and fish, which are the two most distantly related? So looking at this cladogram, we can look at all these junctures at which divergent evolution occurred. So at this point, for example, one group of organisms decided to stay underwater, but some tetrapod groups evolved to live on land, right, and then went along this way. So what I'm asking you to look at is which organisms are the most distantly related. And the answer is sharks and whales. So sharks are all the way over here. They diverged way back here. And then whales took a long time to come up, right? So this tells us that sharks and whales are very distantly related. We can also look at embryonic development patterns to provide more information of our evolutionary past. And anatomical similarities are often most obvious in embryos, because all these embryos share similar developmental processes. And we can look at these two structures. These are two skulls of a chimpanzee fetus compared to a human fetus. So as embryos, these two skulls look very, very similar. But the two species follow different developmental pathways. And these grids superimposed on the skulls show the relative growth of each part of the skull at different points in development for each the chimpanzee and the human. So for example, in an adult human, the brain is always going to grow more so than it would in a chimpanzee, where the jaw of a chimpanzee is always going to be larger than that in a human, right? So different structures become more apparent as development proceeds. As embryos, right, let's look at the earliest embryo. A snake, a chicken, a possum, a cat, a bat, and a human don't look that different from each other, right? Especially with an untrained eye, you would think that these are human embryos. It's only in the late stages of development that you see more distinguishing features between the organisms. But again, if all of these different unrelated, but if all of these organisms look the same at the earliest point in development, wouldn't that be evidence for evolution from a common ancestor? Right? So again, by looking at these, it's hard to refute that we have commonalities with a bat when we look so similar to a bat in the very early stages of development, or even a snake. Right, so again, this also provides evidence of evolution. There's a certain group of genes called homeotic genes that control embryonic development. And these genes dictate the organism's body plan. So it's really interesting because these are conserved among all organisms, from fruit fly to a mouse to a human. So on a fly chromosome, there is a linear arrangement of different genes that specify where certain structures should be placed in the body. And interestingly, in mice, there is a great overlap. There is homology between these genes that dictate the mouse body plan. Similarly, there's an overlap between the the fruit fly body plan and the human body plan. So all of these genes on a fly specify the position of head, thorax, tail. And we have very similar genes on our chromosomes in humans that specify where the head should be, where the lumbar region should be, etc. So these are called homeotic genes. Many homeotic genes that control body plan are called Hox genes. And Hox genes are master genes that encode transcription factors that can turn on the expression of other genes. This could get a little tricky. But recall that every single cell in this fly has antenna genes and leg genes. All cells are the same. But how does the fly know where to put its legs and where to put its antennas? Right? If every single cell has the same instructions, how does this part of the fly know to be a head and to form antenna, and how does this part know to form legs? Well, it turns out that there are Hox genes that encode transcription factors that are only expressed in different parts of the body. So this transcription factor is only present in the abdomen, is only expressed in the abdomen of the fly. So it will tell all the abdomen genes to be expressed only over here. Of course, there are abdomen genes over here, but this transcription factor is not expressed over here. So the abdomen would not be developed over here. So there's actually an arrangement on the chromosome of the different Hox genes, and that correspond, the different colors correspond to the portion of the fly that it could control. So you can see this part, UBX is a transcription factor that controls the midsection of the fly. If I switch the order of these genes, I would mess up the entire body plan. So this fruit fly has a mutation in a Hox gene that controls where the legs develop, and here I put the leg transcription factor where the antenna should be. So now legs grew instead of the antenna. Right, because this is telling, if I put leg transcription factors over here, it's going to tell these cells to form legs. So what's happening really are, again, these Hox genes get only expressed in different parts of the organism, and those will activate different genes, saying, okay, these guys by the head should turn on the antenna genes. These guys over here should form the leg genes. So these control aspects of development, and these are conserved between flies, the insects, and mammals like us. And evolutionary developmental biology, also known as Evo-Devo, is a field of biological research that compares the developmental processes of different organisms to try to figure out the ancestral relationships between them. So you look at how different organisms develop, and then you could basically figure out what they have in common and how their differences came about. And there's a really, there's a funny video that was put out. I'd like you to watch the whole thing, or part of it at least. I'll show you a little bit. But this kind of talks about Evo-Devo, evolutionary developmental biology, in a musical way. So let's watch a little bit of this, but you should watch the whole thing on your own.
Hi. Still divide and decide on 1,000 fates. Did you ever figure how they know? Be back. We are built of modules combined in a planned out way. Each new piece must be told where to go. Oh, now there's a science helping us to understand how our cells encode this architectural plan, signaling each other with genetic tools. Oh yeah, wow. Phenotype the interface for mouse and man, genotype the files and the sub-programs. What then are the switches, circuit boards, and root code? Evo-Devo, looking at the logic in the ways that we grow. Every gene directed by a signal key code. The proteins that can activate, enhance, or veto. Evo-Devo, the signals are controlled by other genes that signal, calculating in a network labyrinth, where the heart and liver and the hands and feet go. Signal mapping to this region where the activity goes. With circuits so deeply built upon, they're older than the million. The millions awake every day. In a crucial pathway, changes tend to get torpedoed. Where they go, calamity goes. As the circulation goes, see down they cascade like a.
So that's a little bit. The video is very clever, has a lot of scientific references in it that are very accurate. So that was Evo Devo, evolutionary developmental biology. So sometimes we can get variations in gene expression from changes in non-coding DNA, not necessarily the genes themselves. So we know that there's only a limited amount of genes that actually encode for proteins. But there are non-coding genes, right? There are non-coding DNA, like promoters and enhancers, that can control when and where different genes get expressed. So I want to give you a simple example. In this fruit fly, there's a gene called yellow that produces black pigment. And normally, the black pigment is turned on in the abdomen because there are certain transcription factors present in the abdomen that turn on the yellow gene, making this part black. However, suppose there's a mutation in an enhancer in the wings. There's a change in the DNA randomly that allows a transcription factor to bind and activate the expression of the yellow gene in the wings. This was never done before, right? This is not supposed to happen, quote unquote. But this is basically turning on the pigment gene in the wings, where it was never allowed to do that. There could also be a mutation that doesn't allow this transcription factor to bind to the abdomen anymore. And that would cause a fly to form without any abdominal band. So again, just by controlling regions that are outside of the genes, we can control whether or not a gene will be expressed and when and where it will be expressed. So this is what contributes to a lot of the diversity we see, right? You don't have to actually change the genes themselves. You can change the way that they're expressed. And that contributes to a lot of variation in the biological world. So up until this point, we discussed fossil evidence and comparative anatomy, and we saw that there's lots of convincing evidence for evolution. So 50 years ago, that was pretty much the evidence we had, and it was already pretty convincing, right? When you look at the comparison of embryology, and you look at the fossil record, there's a lot of evidence. However, now that we understand how to sequence DNA, and we understand how to sequence proteins, and we can look at the amino acid sequences, we have more proof than ever that we evolved from a common ancestor. So comparing DNA and protein sequences can determine evolutionary relationships in very specific detail. Right. It is very highly unlikely that two unrelated species would evolve exactly the same DNA and protein sequences just by chance. Right? You would imagine that they have a common ancestor. Just like you would say, if you discovered the Spanish language and the Italian language, you wouldn't assume that both of those were completely randomly made up independently, because there's so much commonality between the two languages and the roots and the alphabet that you know there's a common ancestor, right? They both come from Latin. Right, so the same way we can look at our genes, and we have the exact same amylase gene that breaks down starch as do chimps. We just have an extra copy of it because we eat more starch than chimps do. So again, this is conclusive evidence that we share common ancestors because we're looking at the instructions that make us who we are, and other organisms have those same exact instructions. So we can't be that unique after all. A way to look at how related two organisms are to look at their similarity in protein sequences. So for example, all organisms have a protein called cytochrome C. I should say all eukaryotes. All eukaryotes have a protein called cytochrome C in their mitochondria. Every single eukaryote from a yeast to humans have this protein. But we can look at the sequence of amino acids in cytochrome C and look at the number of differences between humans and other organisms and figure out how distantly related we are. The more amino acid differences between the species, the more distant the common ancestor. So for example, we have 40 amino acid differences in our cytochrome C protein as yeast do. And if we only have one amino acid difference between rhesus monkeys' cytochrome C and humans, that means we're a lot more related to rhesus monkeys than we are to yeast. So we can look at these amino acid differences and infer how distantly related two organisms are, right? Because the more differences they have, the more distant they must be. We can also look at DNA. So that was looking at amino acid sequences, but we can also look at DNA sequences, and we can study evolutionary relationships. We can also determine the relative dates of evolutionary events by looking at what we call molecular clocks. Molecular clocks. So a molecular clock uses DNA sequences to estimate the time when organisms diverged from a common ancestor. So DNA accumulates mutations at a regular rate. So we know roughly that there's approximately, actually it depends on the species, but we can say that in humans, we differ in about 5% of our nucleotides with chimps. So 5% of our nucleotides are different from chimpanzees. And we know that nucleotide substitutions occur at roughly 1% every million of years, right? Just DNA mutates at a regular rate. Just like isotopes decay, so do DNA mutations occur at a regular rate. So we can use these rates to infer how far along two organisms diverged. So like I mentioned, chimps have about 5% differences in their DNA sequences as humans. And we know that all DNA gets substituted and mutates at about 1% every million years. So if there are 5% differences, that means that our DNA sequences must have diverged from chimps roughly 5 million years ago. Again, this is an estimate. But again, if another example, if a gene is estimated to mutate only once every 25 million years, then if you only have two differences, then that must have been 50 million years passing. And we can look, and we can compare two modern species to a common ancestor and kind of piece back which was the first mutation to occur and at which point these mutations occurred. So again, it's not as specific because mutations can occur due to environmental conditions, like if there's extreme UV radiation, then mutations can happen more frequently than not. But roughly speaking, we can estimate the relationship between two organisms based on looking at their DNA sequences and their differences in DNA sequence. So that's called the molecular clock. So you would have to know the substitution rate between the two in that organism. And then you'd be able to look at, you could say, okay, if I have two substitutions, and I know that substitutions occur this many times per year, then this many years must have passed. Right, so that's what the molecular clock tells us. So to conclude, we said that evolution has plenty of evidence to support it, and we can study several different fields to provide evidence for the theory of evolution. We first spoke about fossils, and we could figure out the age of fossils using relative dating or radiometric dating, which is called absolute dating. We could also look at biogeography, looking at fossils and how they distributed along Earth's continents. Right? We could figure out what Earth might have looked like millions of years ago. We can also look at comparative anatomy and see how we have homologous structures to other organisms. We also looked at embryonic development, seeing how similar organisms are in their early stages of development, which also is evidence for evolution. And finally, we discussed how life's molecules, like DNA and proteins, can tell us evidence of evolution, and specifically, protein sequences can tell us the degree of relatedness between two organisms, and DNA sequences can tell us how far off how far along two species diverge from each other, based on knowing how frequently mutations occur in the genome. So this is a good video contained in the ebook that goes over all the different aspects that we spoke about, kind of one by one. This is a very good video that you must watch. Gives you a very good overview of everything. And that's all I'm going to talk about for this. So thank you for listening, and I hope you learned a lot about evolution and are confident that the evidence supports it. I hope you have a great day.